Oh my
All right, guys. Thank you.
Yeah.
So good to see you. Here's what we're going to do this hour. We're going to just do a little bit of review of chapter 21 and 22 concepts, because I think it's worth going back and reviewing those since the quiz is on that. I actually forgot to make copies of the quiz. I'm sure you're very disappointed in that. So we're going to have to do the quiz tomorrow. I'm not disappointed.
I'm disappointed, Mr. Z.
All right.
I was all prepared.
On a serious note, though, we're going to spend the first maybe 15 to 20 minutes just reviewing chapter 21 and 22 concepts. And hint, hint, whatever I emphasize, you might want to study. Wink, wink. Okay?
Yes, sir.
And so having your chapter 21 and 22 notes out might be helpful, I suppose. And then after that, I'm going to introduce chapter 24 to you. I know you guys have already started reading chapter 24, and that active reading guide is due tomorrow. It's really short. It's not even 4 pages. It's more like 3.5. But anyway, first, I want to go over with you the idea of gene density for a minute. There it is. I kind of breezed over this too quickly, so we're going to camp on this for a second. Notice that if we compare different species like bacteria, various kinds of eukaryotes, we want to find out what the gene density is for these different species. Yes. Oh, this right here is chapter 21. Yep, sorry about that. Chapter 21. So, see this column where it says number of genes, and then you see this column here where it says genes per megabase. The MB stands for megabase. A megabase is basically 1,000 bases.
It's a million.
Sorry, million. Thank you. Yeah. I'm really good at sounding like I know what I talk about.
This is on the bottom. That's the only reason I did it.
Yeah, right. Gigabyte, megabyte, thank you. Megabase. Million. So, which species has the highest gene density? Just take a close look at this table for a minute. All right, so pay really close attention to the second column because that's number of genes. And so here, right here, yep.
Rice has the most amount of genes?
Right, in terms of raw number of genes, yep. But then, remember that not all of the bases of DNA comprise genes that code for proteins. Some bases actually are part of the introns, or maybe they are regulatory sequences. So in this third column where it says genes per megabase, or in other words, genes per 1,000 million bases, you're getting that wrong. This is the third column, and this is going to show us gene density. So which species seems to have the highest gene density? The highest gene density. In other words, look at the third column.
The third one.
Yeah. So 1130, right? 1,130 genes per million bases. Now, let's think about this. Which one has the least gene density? In other words, the fewest number of protein-coding genes.
Humans.
Per megabase.
Oh wait, or is it the lily?
Homo sapiens, unless the lily family plant technically has more, we didn't determine that.
So you see this number 7? That means for 1 million bases in the human genome, you've got on average only 7 genes in those 1 million bases. So what that means is that humans have the lowest density of genes. Yes, Carla.
Does that mean we have the lowest variability, like variation?
Not necessarily, but what about all those other non-protein coding bases of DNA? What are they used for? Regulation mainly, very good. So this means that in humans there's a lot of DNA that's involved in regulating the expression of those protein coding genes, whereas in other species the amount of regulation seems to be less. At least that's what this data seems to be indicating to us. Yes?
So is the third column saying for every 1 million base pairs, there are only 7 genes?
On average. That's
in
my
humans.
understanding.
Okay, I just want to make sure I was understanding you correct.
Yep. Very good. All right, good. So,
Is that because we have so many megabases?
well,
Like we have the most megabases than any other.
we do.
organism right now.
And maybe one reason we have so many is simply because we have a need for all these regulatory sequences that control which of those genes will be switched on, which ones will be switched off. So it's very elaborate, and I've taught you, of course, the mechanisms by which those genes are switched on and switched off. And those mechanisms are more complicated and more involved in humans. We could probably reason that that's the case looking at this data. Yes?
What are you emphasizing here?
What am I emphasizing? Which species has the lowest gene density and thus has the greatest number of regulatory sequences in its DNA? Which species would that be?
So we regulate.
We, meaning humans, yes.
Regulate.
Yeah. Now, all organisms have regulatory sequences. Don't misunderstand me on that. But humans have evidently a lot of that.
So does that mean
Yes.
Archaea, Globus, Fulgus? Like, okay, I'm not saying they don't regulate, but they just regulate a lot less than us.
Compared to us, they have fewer regulatory sequences, correct? All right, good. All right, moving on. I wanted to go over with you transposons and retrotransposons. You might remember this. Okay, so remember there was this lady named McClintock, and she studied corn, and she learned that some genes had the ability to, it seems, jump from one part of a chromosome to another, although they don't literally jump. But there are a couple different ways that they move. They could either be, a section could be replicated, and then that replicated section of DNA could transpose, or I should say move to another part of the chromosome. And that is shown, and that's what I just mentioned here, the transposons. So here's how it works. Remember that if a gene is to, quote, jump, this is how it works. A section of DNA will get copied, so that gene is copied into a replicated molecule of DNA. And then that replicated section of DNA will be transported somewhere else along the length of the chromosome. So now we've got two copies of that gene. Can anybody think of an advantage of having two copies of this gene along the length of the chromosome? Yes, Hayden?
In case something bad happens to one of the copies, you can replace it or replicate.
I never thought about that. What if this gene over here mutates in an unfavorable way? At least we have a favorable copy already.
Yeah.
All right, good. Yeah, so that's a good reason. And if we want more of that certain desired protein, whatever that gene codes for, then this might be an advantage. Then there's what's called retrotransposons. Can anybody remember what the difference is? Yes, Ashley? Or Angel? I don't know why I called you Ashley.
This one takes like the DNA or the part he wants out and puts it somewhere else instead of creating a copy. So there's not two of them, it's just the same one.
Same one? Same one of what?
Same one of whatever it's copying isn't.
Okay, sort of. And you notice there's RNA here. So instead of just copying one gene into another DNA fragment, instead we're actually transcribing that DNA into RNA, and then an enzyme known as reverse transcriptase comes along and converts that RNA back into DNA, but it does so downstream or upstream, and so we now have a new copy of DNA upstream or downstream. But the intermediate is RNA, so that's the difference between transposons and retrotransposons. Hannah.
Wait, what's this like? What would be the purpose of making it into RNA back? Like, wouldn't that be?
Good question. Good question. Hannah asked, "What's the purpose? What's the advantage of retrotransposons versus transposons?" I'm not totally sure. I'm not sure if anybody knows. It seems a little more complicated. So you have to have RNA and RNA polymerase rather than DNA polymerase for this to happen. Yes, Max?
So transposons use transposase, and this one just uses the RNA.
Good question. So this process uses retrotranscriptase, which takes the RNA intermediate and converts it back into DNA so that it can be inserted. So that's a fundamental difference you need to know.
But the original strand is still there.
Yeah, so the original DNA is still there. It remains. Is that what you're asking?
Is that for both?
Yes. So the original DNA also remains here in this process as well. Yep. Good. But I'm sorry, I don't know the answer to your question, Hannah. We just know that these two processes happen, but we don't know for sure what the apparent advantage of either one is over the other. Yes, Max?
After it copies the DNA from the original site and then where is it inserting it?
Where is it inserting it? Somewhere along the length of the chromosome. And so there are enzymes that cut in certain places and make it possible for this DNA to be inserted at a specific location. When you say where, I can't give a specific answer because it depends on the type of gene, kind of chromosome, the species that's involved. But this little mark here means that there may be a lot of DNA in between this area and that area. We're just not showing it. So somewhere downstream or upstream along the length of the chromosome. Does that help? Okay. Yes.
So for both of them, how many copies of the DNA is there? Because for the transposase, it copies the segment and then it pastes it later. So it's like a copy and paste mechanism.
Yeah, it is.
So there can be
That's right. 2
or more depending on how many copies they want to produce.
Sure. In theory, maybe this gene could be copied and duplicated in multiple areas along the length.
I was gonna say I don't know how many of them, but it would have more than 2 of that code.
It could at
Okay.
least 2. By the time this process is done, we'll have at least 2 copies of that gene. We could have more, I suppose. Yeah.
So then for retrotransposase, how many copies, quote unquote, of that sequence would we have?
I'd say 2 or more, because once this process is complete, maybe the process will repeat itself, and we'll have another copy of that genetic information somewhere else. I don't know the answer to your question for sure, but I don't see a reason why it couldn't happen more than once. It's a good question. Yes, Kylan?
So is the only difference between a retrotransposon and a transposon the way it gets its copy? Are the copies, say took the same exact thing, and one was a transposon, one was a retrotransposon, would they have the same copy, just a different way of getting there?
Yes.
It's if you took away all the bottom part,
if you did the before and after, it would look exactly the same.
Yeah, my understanding is what we're starting with and then what we're ending with is exactly the same, but the process by which we achieve that result is different.
So transposase is clicking the command C button, and then command, and pasting it, and then the retrotransposase is selecting all of it, and then hitting, and then clicking on the mouse and making it copy. And
Yes.
There's
In
more
addition,
steps.
in addition, there's transcribing going on in
I'm just trying
this
to
process.
explain it to myself.
All right, Samantha.
I know when we did retroviruses, it was really that it had a lot of errors because there was no subject. There's a subject for this.
There is, but it's not 100% accurate. Good question.
So would the first one be a safer?
Oh, that's a really good question. Wow, you're definitely thinking outside the box, Samantha. We know that we know about RNA viruses that use the reverse transcriptase and how they mutate more quickly because they don't have this self-correcting replicating mechanism. Your theory is valid in that maybe there's more fidelity in terms of the genetic information being preserved and not mutating very quickly with this kind of mechanism, as opposed to here where we have this enzyme reverse transcriptase. It may not be as accurate. I don't know. Don't know the answer to your question, but it's a very interesting hypothesis. All right, cool. Any other questions about that? Yes, Hayden.
So me and Ashley here were conversating, and we were just kind of confused on why it's supposed to be. It's copy, paste, then cut and paste, but it doesn't actually cut it.
True.
When we did the active reading guide, that's why I said that, because it said it was cut and paste and copy and paste, just like that. But it doesn't cut anything out. It just
What I see here is more of a copy and paste. So maybe cut and paste in this case is not the best description. I see what you're saying there. Yeah. Let's call it copy and paste.
for both.
Yeah.
Okay.
Yep. Of course, the copying process is a little different here because we're actually transcribing into RNA. But yeah. All right, cool. Let's talk about homologies. Homologies. What's a homology?
The same.
The same? Same what?
Same chromosome.
Okay. Well, there are similarities between chromosomes of different species. We could call those chromosomal homologies. So you're onto something there. Yes. What other kind of homologies are there, though?
Let's say wings that are similar.
Yes. Well.
Okay, let's not say that.
Maybe.
The 5 things on the hands of the mammals.
Oh, good. Yes, we talked about the digits of the hands of humans versus the digits in the paws of a
The wing
cat.
one is a different word, right? 'Cause it's
Right.
not the same.
Yeah, when you talked about wings, like wings of a bird versus wings of an insect, we refer to that as an analogy. So I'm going to go over that real quick with you. Actually, I'm in the wrong slide presentation. So a homology is when you have similar structural characteristics, and then analogy is when you have similar functions. So I'm going to go to slide 32 here. 32. I don't know. I've got it somewhere on here. Anyway, yeah, so like a wing of a bird versus the flipper of a whale, that would be a homology. So the structural similarities are there, but the functions are a little different.
Yeah.
Okay. Whereas with analogies, you have a similar function. Like with the bird wing, you have flight, which is the function, and then with the insect wing, still flight, but the structural dissimilarities are significant. So, for example, the bird may have bones, nerves, muscle tissue in that wing, whereas the insect just has a simple membrane tissue. Okay, so that's considered an analogy. Okay, cool. One more thing. Let's talk about Cuvier and Lyell. Charles Lyell and Cuvier. Anybody remember anything about those guys? Anybody know? Remember?
Mess around with flies.
Well, all right, let's focus on Lyell first. What did Charles Lyell do?
Study fosters.
Okay, well, here he is. Oh, that's Linnaeus, I'm sorry. Here it is. So Charles Lyell, of course, was a geologist. So he came up with this idea of uniformitarianism. So this is the idea that Earth changes, all the geological changes happen rather slowly, and over long periods of time, those slow changes have certain outcomes that we see today. And so emphasis on slow, gradual change, all right? He believed that Earth's history consisted of changes that occur at a consistent pace, whether they be continental changes, continental drift. Even though you have events like volcanic eruptions, you have floods and so on, looking over this vast span of time, we'd imagine that these happen at a rather constant rate. That's his perspective. So Charles Darwin came along and said, Charles, you have the same name as me, therefore you must be brilliant. No. But he said, I like that idea, and I'm going to apply those slow, gradual changes to Earth's structure to biology. And he said that organisms or populations will change gradually over time. So Charles Darwin and Lyell actually talked to each other quite a bit.
Were they alive at the same time?
Yeah, so Charles Lyell was a little older than Darwin. So Darwin was a contemporary. Okay, back to Cuvier. All right, so this is a different geologist. He actually lived about the same time as Charles Lyell. His perspective was that of catastrophism, the idea that Earth and its features tend to go through very sudden changes, and these changes happen in a punctuated pattern across the Earth's history. So this is the opposite of gradualism. So a lot of geologists, biologists, have said, well, maybe there's a compromise between the two. Maybe there has been a lot of gradual change, but then there have been some sudden catastrophic events. Like maybe you've heard of, how there was a comet that landed somewhere in the Gulf of Mexico, eons ago, and they think this is how the dinosaurs went extinct. Well, that would be a catastrophic incident. Cuvier would say that something like that would happen, once in a blue moon, but when it does, it drastically affects not just Earth structure, but also plants and animals and the evolution of life. So you do want to be able to contrast those two ideas. All right, I'm kind of going in reverse here. There was a guy named Lamarck. Oh, no, we can talk about Linnaeus, that's fine. You guys want to know for the quiz the different levels of classification. So kingdom would be the most general category. Each kingdom is divided into several different phyla. A phylum is divided into several different classes. Each class is divided into several different orders, and so on. And then you have families, genus, and species. So species, of course, is the most specific category. All right? So you want to be able to know that order. Any questions? What do you think, Marissa? All right. Something else I want to go over with you, different kinds of selection. We talked about natural selection versus artificial selection. And now Charles Darwin is credited with his idea of natural selection, but of course he wasn't the only guy to be thinking about this idea. Alfred Wallace, who also lived at the same time as Darwin, was thinking about how natural selection may explain some of the diversity of life. And so, but Darwin got published first, and that's why he's more famous for the idea. All right. Before I go to the next slide, what's the difference between artificial and natural selection? Artificial versus natural.
One is man-made.
Jamie?
One is man-made.
Man-made. Which one's man-made?
Artificial.
Correct. Very good. So Charles Darwin thought, hey, if I'm breeding pigeons and I'm able to have some control over what physical traits are more frequent in the next generation, then maybe nature has a way of also having that kind of control. But the mechanism's a little different, because if you're breeding pigeons, you can select whatever trait you think is pretty and is going to be a good selling point to those that want to buy your pigeons. But that's not based on survivalhood, right? What's pretty doesn't necessarily make you survive better, necessarily. In nature, though, it really is all about survival. So nature selects based on the qualification of, can you survive? Do you have the right package of genetic information or physical traits that'll help you survive? So we learned a little bit about, and here's the differences between artificial and natural, so you can review that in your notes. It's there. I wanted to talk about tree of life. Tree of life. We talk about fossils. I know I have a tree of life somewhere in here. Yes. Okay, so here's a diagram. It shows us an arrangement of organisms according to their physical characteristics. Now, when an evolutionary biologist looks at this, they're going to think about ancestry. I mean, your book thinks that way, or the authors of your book think that way. The authors of the questions on the AP exam, I can guarantee you, almost all of them are Darwinian biologists. They believe that a common ancestor links together all of these organisms. Now, why don't we ever see a picture? Why don't we see a picture of an ancestor here, or a picture... Bless you. Why don't we see a picture of an ancestor here, or a picture of an ancestor there? I mean, I think that would be helpful, I suppose. Yes, Kylan?
Well, it doesn't exist. We don't know what that looks like. Well, right now it doesn't exist. Like, we only have the two breaking points.
We don't know what it looks like. Correct. You guys got that? Now, I'm not saying that such an ancestor never existed, but science is not able at this point to reveal what that ancestor would have looked like. So where would we look to try to find such an ancestor? Fossils, that's right. So in the fossil records, there are all kinds of species that have gone extinct, and they're very interesting to study. But the problem is that there are so many missing links, is what they call them. It just means fossils that so far are imaginary and we haven't found them yet in the fossil record. Maybe they're there, maybe they aren't. If they aren't, that would be very problematic. Yes, Tristan?
If there's a fossil that like branched off to make all those things, that would have to have all of those characteristics.
Or a lot of them, sure.
That's like...
That'd be scary.
That's like... a super being. That's like...
It's like Jurassic Park.
Yeah. Good question. What would it look like? Students, that's a good question. we're left to our imaginations.
Well, it doesn't have to have every characteristic.
So-
It just
Well,
has
it might
right?
also just
Possibilities.
have them in the gene sequence,
No,
but
yeah,
not
because
express them.
well, and also it combined with some of the lower ones too, so it could have pulled stuff from the lower ones without. It doesn't have to have everything, just the majority.
Just like flight mistakes. Honestly, any of those things flying.
The one that would probably be the most like that animal would be the very top one because it's such a red.
Yeah. It's like a frog with wings and gills and fur.
All right. Yes.
Going back to natural selection real quick.
Yeah, sure.
If there's like
Angel?
you know like I don't know, like when like the male tries to attract the female or something. Is that still natural selection?
That's a kind of natural selection called sexual selection.
Because like
Yep.
you know how artificial selection we can kind of pick like what's prettier like
Right.
to sell or something, right? The male or the female, right, is picking like they're picking. That part's not like based on survival or fitness. They're picking like the cutest boy out there. So.
Okay. Is that all there is to it though?
Well, probably not. But at the basic level, like, is it still natural selection? Or is sexual selection a branch of natural selection, or is it its own thing?
Correct, it is. Yes. Now, one aspect of sexual selection that's often played out is through dueling, like mule deer, for example. The males will fight for, mating rights,
That's like
and so that's called intrasexual selection. But remember that there's a reason for the dueling process. I mean, why is it beneficial for the males to beat the crap out of each other?
To weed out their threats. So only
Yes.
the strongest can reproduce.
Okay, yeah, that's a pretty good answer. I like it. And also, whoever the winner is has those genes that are associated with strength and speed, those are the traits you want to have to be able to win a fight. And then those genes are passed on if you have the mating rights. And then with intersexual selection, when the female is choosing the male, which in most cases that's the case. With some species, the male chooses the female. But anyway, remember that the female, if she chooses who's colorful or who sings the best or who dances the best, dancing, one could be strongly associated with athleticism, the ability to flee from predators. It's not necessarily all about who's the prettiest or sounds the best. When a female is choosing the male, she's choosing who sounds the best and who has the most appealing dance. But remember, embedded in that is some survival characteristics that would help an organism survive. Does that make sense?
So when we pick up flower to breed with the other one, just for the sake of selling a purple flower instead of a blue one, that's artificial because we're not doing. It has nothing to do with like us wanting to make the flower survive.
True. That's true. On the other hand, if a honeybee chooses to pick up pollen or get nectar from, let's say, the white flower instead of the red one, well, the white flowers might have more reproductive success. That's not sexual selection; it's natural selection. Is there a survival advantage to being white versus red? I don't know, but we're still going to call that natural selection because there's nature choosing what's going to reproduce. Good question. All right, good. I think we've covered just about everything we need to.